Good morning, everyone. Welcome to another one of the COVID-19 webinars brought to you by the National Institute for Occupational Health, the NIOH. This, I believe, is our 97th webinar since March 2020, and we have reached over 50,000 participants and attendees in the webinar series. Today's webinar series, I think, is of absolute great importance and of great interest to the stakeholder audience of the NIOH. And I just note that the IT colleagues, Tabani and Glenn, has indicated that we've had a bumper registration for this session, over 1,600 people that have registered. However, our webinar platform can only accommodate a maximum of 1,000 attendees logged in. And after that, once people can't join after the 1,000, we will be encouraging them to go to the live YouTube streaming of this webinar. And to the question asked by one of our attendees in the question and answer box, if I can quickly just pop that up again, I had to move it. Apologies, Annalie Wickham. Yes, the session is being recorded, and all recordings, audio, video, and presentations of the presenters will be shared via link uploaded to our website, which is zero rated. Okay, so that's just getting that quickly out of the way, Annalie. And a bit of quick admin. Do not use the question and answer box. That's Annalie, Susan, and Charmaine for, sorry, not Susan, for asking questions about admin, CPD points, certificates, and so on and so on. That you must use the chat box. Chat box is for general administration, as well as for any questions you have related to aspects of CPD certificates and recordings and so on. I'm going to encourage you to use the question and answer box, the Q&A box, to ask questions, all of your questions. No questions in the chat box, all your questions in the question and answer box for our presenters, and they will be encouraged and asked to just answer your questions there in the question and answer box. We also have a number of panelists from the occupational medicine section here at the NIH, led by Dr. Nompumelelo Ndaba, who will be answering those questions for you there. The raised hand function is not valid for such a large number of attendees and participants. So I'm going to ask you not to use the raised hand function, but to type your questions in the question and answer box or general aspects in the chat box. So Zanele Gwenya, if you could just lower your hand, that will be much appreciated. So just for the formal start, greetings everyone. A very warm welcome, as I said, to another webinar in the COVID-19 series brought to you by the National Institute of Occupational Health. The NIH is a specialized division of the National Health Laboratory Services, the NHLS. And so we're delighted to have you here on this particular topic of COVID-19 vaccines and the workplace. And we've got three distinguished guests who's going to share their presentations, thoughts, experience, and expertise with you, and as well as answer your questions in the question answer box. And towards the end, we'll have a Q&A session to focus on the key questions asked, the key observations that our speakers and our presenters have made, not just from your questions, but from the different contributions they've made, and we will engage in that last session. So on the topic of COVID-19 vaccine safety, we've got Dr. Desiki Kalonji, and Dr. Kalonji will deal with that particular topic. A second after that will be followed by COVID-19 workplace vaccination programs, and that's dealt with by Dr. Yamanya Tembo. And thirdly, finally, on COVID-19 vaccine update, and I'm sure a lot of us are looking forward to this as well, by Professor Shabir Madi from WITS. So without further ado, I'm going to ask, I just want to check if Dr. Singh is with us. I've now unfortunately forgotten to just check if she's here with us. So it doesn't seem like it is the case. I'm therefore going to ask Dr. Nompobelelo Ndaba, and this is off the cuff, just to do a welcome in the absence of Dr. Singh. Dr. Tanisha Singh is the head of our COVID-19 committee here, known as the COVID-19 Occupational Outbreak Response Team, the OO team. And unfortunately, she's not in a position now to assist us today. And I'm going to ask Dr. Ndaba, as the head of the occupational medicine section, just to say a few words of welcome. Over to you, Dr. Ndaba.
Good afternoon, colleagues. On behalf of the NIH OHOT task team, and on behalf of the occupational medicine section, we welcome you to this webinar. We know it's long been requested. We have long been thinking we are going to host this. Special welcome to our guest speakers today and the presenters. We are hoping to gain a lot from today's presentation, and we also have panelists who are going to be assisting us with questions. We hope to have fun and learn from this. And special welcome to Professor Shabir Madi and the other speakers, really for making time in their very busy schedule and prioritizing us at this time. Thank you. Welcome on board. Thanks.
Thank you very much for that, Dr. Ndaba, for rescuing me there for a moment. So without further more details on this, I'm going to quickly bring up the PowerPoint slide in order to introduce our first speaker by way of introduction, and to ask her just to prepare to share her slides and to open her microphone, Dr. Kalonji. So just to quickly share this particular slide, one moment. There we go. So I'm not always good at the slide sharing, but I hope that that's visible to you. Maximized just a little bit. Okay, so by way of introduction, Dr. Desiki Jenny Kalonji is a qualified medical doctor with additional specializations and an MEd from the University of KwaZulu-Natal. She's a clinical research site leader at the Isipingo Research Site, the HIGRU SAMRC in Durban. That's the South African Medical Research Council. Dr. Kalonji is also a principal investigator for 2 HIV prevention trials, 1 PrEP and 1 HIV vaccine, and 2 COVID-19 trials the COVPN 3008 and Sisonke 4. As the CRS leader, Dr. Golonji have oversight of all studies at the site. Isipingo CRS is experienced in all phases of clinical trials, phase 1 to 4. The site has experience with both HIV vaccine and SARS-CoV-2 vaccine trials and vaccine safety. As a principal investigator, vaccine safety is her bread and butter. And with that very short introduction, I ask Dr. Golonji to join us and share her presentation slides. A quick admin reminder while that's happening to all of our attendees, please type your questions. That's questions related to the presentations and the subject matter of our speakers and presenters in the Q&A box. No admin in the Q&A box. All admin related matters is for the chat box. We will monitor that, and we want to focus mainly on the questions that is happening in the chat box. To Bulewa and others, please do not raise your hands. You will not be allowed to open your microphone. Thank you very much. And over to you, Dr. Golonji. I see your slides are maximized, and just to check, your microphone is open. I hand over to you.
Good afternoon, everyone. Thank you for the invite. So my name is Dr. Tshiki Jenny Kalonji, and as seen in my bio sketch, so I'm pretty much a principal investigator, and so vaccine safety is quite important in my line of work. So I'm just
apologies for interrupting, Dr. Kalonji. Could you just move closer to your microphone? Thank you.
Can you hear me better now?
It sounds a little bit better, thanks.
All right, okay. So I'm going to go straight into my presentation. Thank you. So the content of my presentation includes vaccine safety, then we'll talk about some adverse events, vaccine safety in immunization programs, pre-licensure vaccine safety, post-licensure vaccine safety, and also a little bit on vaccine safety regulations. So vaccine safety is actually defined as a process that maintains the highest efficacy of and lowest adverse reaction to a vaccine by addressing its production, storage, and handling. Vaccine safety is part of immunization safety. And as you know, with all medicines, every vaccine needs to go through extensive and rigorous testing before it can be introduced in a country. Once they are in use, vaccines must be continuously monitored to make sure they are safe for the people who receive them. And it can be classified as pre-licensure and post-licensure. Just to discuss some expectations towards safety of vaccines, vaccines are not completely risk-free. Adverse events do occur. Thank goodness most adverse events are actually minor, such as redness at injection site or fever. There are more serious reactions, but these are uncommon and sometimes very rare, and these include seizures and anaphylaxis. The general public has a very low tolerance to any adverse events following vaccination. And simply because vaccines are given to healthy persons to prevent disease, so a higher standard of safety is expected of immunizations compared to medication that are used as treatment. There is a greater need to detect and investigate any adverse event following immunization than is generally expected for other pharmaceutical products. And this is just a diagram to kind of show that how the public has a low public tolerance, and therefore it is a very important responsibility of national regulatory authorities to ensure with rigor the quality, safety, and effectiveness of vaccines and pharmaceutical products. Now, just a little bit about adverse events. So an adverse event following immunization is any untoward medical occurrence which follows immunization. It does not necessarily have a causal relationship with the usage of the vaccine, so we describe them as related or unrelated, and these can actually be divided in 5 categories. There are vaccine product-related reactions, which is actually due to inherent properties of the vaccine product. There are immunization error-related reactions, which is caused by inappropriate vaccine handling, prescribing, or administration. Then you've got vaccine quality defect related reactions, which is due to any quality defects of the product. There's immunization anxiety-related reactions, and that arises from anxiety about the immunization and the process and the fear of injections. There are also what we call coincidental events, which is any event that happens after the vaccination but is not caused by the vaccine or the vaccination process. And all of these different kind of adverse events following immunization have implications when it comes to COVID-19. Causes of adverse events. Vaccines contain different components to make them effective. And technically speaking, each component in a vaccine adds a potential risk of an adverse reaction. Regulatory authority must ensure that all vaccine components, singly or and in combination, do not compromise vaccine safety. It's also prepared with different types of antigens using different scientific methods, such as inactivation, attenuation. In addition, some vaccines include components to enhance the immune response, such as adjuvants and conjugated proteins. And vaccine can also include antibiotics and stabilizers to reduce contamination during the manufacturing process. And for this reason, manufacturers usually recommend the route of administration that limits best adverse reactions of the respective vaccine. Under ideal conditions, vaccines should cause no adverse events and completely prevent the infection that they target. Then again, this is an ideal situation and it does not exactly happen. The key, therefore, is to minimize as much as possible adverse events and ensure a safe use of vaccines. So this is just a table outlining the different kind of adverse events and how common and how frequent they are. So for example, you've got very common, which has normally some local adverse events. Then there are some that are uncommon and some that are rare and some that are very, very rare. When it comes to COVID-19 vaccines, common side effects happen locally, for example, pain, redness, and swelling at the site of injection. And then there are also some systemic adverse events such as tiredness, headache, muscle pains, fever, nausea. There's another special group of adverse events that we call adverse events of special interest. And this is a pre-identified and predefined medically significant event that has the potential to be causally associated with a vaccine product. And this needs to be carefully monitored and confirmed by further specific studies. And we call them short AESIs. Here's a table of AESIs specific to COVID-19. As you can see there, it includes coagulation disorders. I'm sure you're all aware of the extensive media coverage regarding coagulation disorders and defects that came along with COVID-19 vaccines. Now, there needs to be a balance between efficacy and safety. So vaccine efficacy refers to the ability of a vaccine to bring about its intended beneficial effects on vaccinated individuals, and of course, in a defined population and under ideal conditions of use, which is why in clinical trials, we talk about efficacy, and post-marketing, we tend to discuss effectiveness once it is in the market. So the potential benefits of an effective vaccine must actually be weighed against the potential risk of an adverse event following immunization. Vaccine-associated risk is the probability of an adverse or an unwanted outcome occurring and the severity of the resulting harm to the health of the vaccinated individuals in a defined population and again, under ideal conditions of use. An important criterion for vaccine safety that regulatory authorities must establish is this risk-benefit assessment of immunization with a particular vaccine in a defined population. Public confidence in vaccine safety is increased by clear communication of this risk-benefit assessments, comparing the very low vaccine-associated risk with the very significant benefits of vaccination. So the greater the gap, the better. Risk-benefit assessments should be applied to most situations relating to the efficacy or safety of vaccines to ensure public safety and public health. Now, there is a special scenario I just wanted to discuss, which is vaccine safety in immunization programs. There is a graph that will follow. So I just want you to imagine different stages. There is a pre-vaccine era, for example, and in a pre-vaccine era, you have a new infectious disease, like for example, SARS-CoV-2, and it results in high incidence of disease, high morbidity, high mortality at times. And at the time, before the vaccine, there is no vaccine, and therefore there's also no adverse events. And this pre-vaccine stage can be considered as a stage 1, the phase before a vaccine is introduced. In stage 2, an effective vaccine is introduced to prevent a particular disease. And then there is an increase in immunization uptake and therefore a decrease in disease incidence. But at the same time, there's a new vaccine and therefore there's an increase in adverse events following immunization, whether it's real or perceived. Paradoxically, it is just when vaccine benefits are most apparent and vaccine coverage is at its highest that vaccine safety concerns are also most likely to increase in the general public. So now there's an increased focus on adverse events following immunization, and this is often, and you have seen it with COVID-19, intensified by media coverage of one or a few case reports, and this can lead to a loss of confidence in the vaccine by the public, and also a reduction in vaccine coverage. So the more successful a vaccination campaign is, the less visible the prevented disease may become to the public. And as the threat of the original disease vanishes in the perception of the public, the attention is mainly focused on adverse events of the vaccine. And then it can lead to a distorted perception of the risk of the vaccines and also a negligence of the much greater health threat by the original disease, which may lead to decreased acceptance of the vaccine. And this leads us to stage 3, which is a resurgence of the disease to higher or sometimes even epidemic levels. And this resurgence of the disease or an introduction of another alternative vaccine can result in renewed public acceptance of vaccination against the disease. And this leads to stage 4, where vaccination levels increase and the disease is reduced to earlier low levels. And if we are lucky, for certain vaccine preventable diseases such as smallpox that can be eradicated, then vaccine use can stop completely and thereby removing any risk of any adverse event resulting from its use. And that would be considered stage 5. Now, to ensure that the cycle does not repeat, it's very important that any vaccine safety issue requires timely detection, evaluation, and response efforts to gain and maintain high public confidence. So this is what the graph looks like. You can see stage 1, pre-vaccine, a lot of disease and little vaccine coverage until a vaccine is introduced. And that would be in stage 2, where you've got increased coverage and the disease incidence decreases. Stage 3, you've got loss of confidence. And again, we start seeing outbreaks. And with stage 4, there's a resumption in confidence. And again, vaccine coverage increases and disease incidence decreases further. And if we're lucky and there's a stage 5 and eradication, then there's no more disease. And at the same time, we can phase out the vaccine. Now we're just going to discuss pre-licensure vaccine safety and post-licensure vaccine safety. So vaccines undergo extensive testing and review for safety, for immunogenicity and efficacy. It starts in the lab, and then in animals, and in 3 phases of clinical trials in human subjects. And this is before licensure, before it goes onto the market. Monitoring adverse vaccine reaction is a major safety component of pre-licensure clinical trials. In my line of work as principal investigators, this is pretty much what we do the entire day. Now, pre-licensure studies often identify common and acute negative reactions that occur with a frequency greater than 1 in 10,000 vaccinations. And that really depends on the sample size of the study. But the sensitivity of detection of uncommon and very rare adverse events are those with delayed onset is however low in these pre-licensure trials. As a result, continuous post-licensure monitoring of vaccine safety is needed to identify and evaluate such adverse events. So here we go, and we talk about the 3 clinical phases pre-licensure. There's phase 1, and phase 1 is really looking at some safety issues, immunogenicity, tolerability, and it's normally low-risk individuals and very, very small studies, like 10 to 100 participants. Phase 2 looks at safety, potential side effects, again, still looking at immune response, and also looking at optimum dosage and schedule. And again, and there you've got an increased number of participants. Clinical trial phase 3s look at clinical efficacy in the disease prevention and provide more safety information. And there you can have large sample sizes of participants. After this, there's a submission where the vaccine application is submitted to regulatory authorities for approval for the market. And if this is approved, then the vaccine is introduced in society. And before I move on to post-licensure, I just want to discuss this emergency use listing, which I think you've seen a lot of with these COVID-19 vaccines. So this emergency use listing, it's a unique WHO facilitated regulatory pathway. It can only be used in a declared public health emergency of international concern or other public health emergency designated by the WHO Director General. And this emergency scenario actually allows for a product to be listed while it's still unlicensed, based on an earlier package of safety and efficacy data than is the norm. And the first vaccine to actually be submitted to the WHO EUL under the revised procedure was the novel oral polio vaccine type 2 in 2019-2020. And this vaccine was developed to better address the evolving risk of type 2 circulating vaccine-derived poliovirus. And this was elicited as a declaration of PHEIC by the WHO in 2014. And the EUL actually plays a critical role in accelerating equitable access to COVID-19 vaccines, enabling manufacturing countries to use an emergency pathway to authorize products. So the norm for pre-licensure vaccine safety, if you look at the trials, is that you can see that it takes many years, many years. If you look at from the beginning to a phase 1, 3 to 8 years, from phase 1 to phase 3, 2 to 10 years, and regulatory review can take 1 to 2 years. So it can take up to 15 years before a vaccine under normal condition reaches the market. But under the EUL pathway, you can see that all these are reduced to months. So what was taking 3 to 8 years is now 2 to 3 months. And then from phase 1 to phase 3, you've got 6 to 12 months, and phase 3, 3 to 9 months. And this is this accelerated vaccine development leading to then EUL submissions. And you may ask yourself, how do they do it? And the good news is that there are no steps that are skipped. But what happens is that phases are overlapped, and this shortens development time. And I can tell you that at a site level, we do feel it. EUL is actually more intense, and there's a lot of work that goes into it, but this is what happens. So this pandemic paradigm requires multiple activities to be conducted, and it is at the financial risk to developers and manufacturers, because they may not necessarily know whether the vaccine candidate will be safe and effective. And also it includes very early manufacturing scale up to commercial scale before establishment of necessarily a clinical proof of concept. But this is how the EUL works. And just to give you a slide that kind of shows of the different vaccines that are in development, you can see that there are many vaccines in preclinical, and then some are in phase 1, phase 2, phase 3, then the number of vaccines that are in use, and then phase 4, which we will discuss now, as that is part of the post-licensure safety. So post-licensure vaccine safety, this involves post-licensure surveillance of vaccine safety, and it's actually quite critical. The conditions and the reasons for safety monitoring now change following licensure and the introduction of a new vaccine in a country. Vaccines are now in use as opposed to being under trial conditions, and they're in use in the general population, and recipients are no longer monitored in clinical trials with these narrow inclusion-exclusion criteria. And all of a sudden, subpopulations that are commonly excluded in clinical trials will get vaccinated. There are large numbers of people are being vaccinated, and other factors that can lead to adverse events following immunization, such as incorrect administration practices, need to be monitored for safety. Uncommon and rare vaccine reactions are now seen, and reactions with delayed onset, which are not detected before vaccines are licensed. So now healthcare providers should understand that vaccines can demonstrate rare and potentially serious adverse events in this post-licensure vaccine safety. In these instances, policymaking bodies have actually judged that the individual and community benefits of vaccination outweigh the risks. And there are several surveillance options for post-licensure vaccine safety. And just to define surveillance, so surveillance is this continual systematic collection of data that are analyzed and disseminated to enable decision-making and action to protect the health of populations. The adverse events following immunization surveillance systems are specific to monitoring adverse events associated with vaccine use. And there are two types. We have passive surveillance systems and there are active surveillance systems. And this is just the surveillance cycle. You can see that the adverse event is detected, then notified, reported, it's investigated, and then there's analysis of data, then there's causality assessment, and then feedback. Now for passive surveillance system, these are actually the very common surveillance systems. They're spontaneous reporting systems. They're the cornerstone of most post-licensure safety monitoring systems because they're relatively easy to implement, their cost and ability to capture unexpected events. You can monitor events reported by healthcare providers and consumers, and they do not actively seek out and collect data or measure outcomes using study protocols. Now, for post-licensure, there are different types. There's this post-licensure clinical trials and phase 4 surveillance studies. And in this case, the vaccines undergo clinical trial to assess the effects of changes in vaccine formulation, vaccine strain, age at vaccination, number and timing of vaccine doses, and also simultaneous administration or interchangeability of vaccines from different manufacturers, for example. And it helps to improve the ability to detect adverse events that are not detected during pre-licensure trials. There are also other surveillance systems such as large linked databases. And these actually, because they enroll populations numbering from thousands to millions, they can detect very rare adverse events. Other ones are such as the CISA, which is in the US, for example, it's tertiary clinical centers that have been used to conduct research on immunization-associated health risks. Now, a little bit about immunization safety surveillance. So immunization safety is the process of ensuring and monitoring the safety of all aspects of immunization. And that includes vaccine quality, adverse events, vaccine storage and handling, administration, disposal of shots, and management of waste. The skills and infrastructure to deal with genuine vaccine adverse reactions are essential to public safety, as well as to prevent or manage fear caused by false or unproven signals from patients and health workers. There's also vaccine pharmacovigilance, and this is defined as a science and activities relating to the detection, assessment, understanding, and communication of adverse events following immunization and other vaccine or immunization-related issues, and to the prevention of untoward effects of the vaccine or immunization. There are special considerations when doing surveillance, such as training of healthcare workers, because healthcare workers administer the vaccines. They're also on the front lines and usually the first responders to adverse events and also adverse events of special interest. They need to be trained on how to detect, report, and respond to adverse events, and also stabilize the patient and communicate with end users, the community, and the media. Also, the need to determine causality, and independent review is also needed. There's a need for independent review of adverse events separate from the immunization program. So causality assessment requires a team of investigators, including immunologists or other experts, depending on the nature of the adverse event. And the team should not directly include officials from the National Immunization Program, as this may be perceived as a conflict of interest, as they are responsible for investigating adverse events related to administration of a vaccine. So just to touch a little bit on vaccine regulations. So today, vaccine regulation includes a range of functions that cover the entire continuum of vaccine development, licensure, and use. And it includes regulatory bodies at national and international level. So this is kind of just a framework that shows how you have the international and also the national. So the national is mainly the National Regulatory Authority, National Immunization Program, and there's a review committee and other support groups. So in South Africa, SAHPRA, which is the South African Health Products Regulatory Authority, is the national regulatory authority that oversees the safety, efficacy, and quality of all medicines in South Africa, registered in South Africa, including vaccines. The National Department of Health Expanded Program of Immunization, that is our national immunization program for COVID-19 vaccination programs, and they collaborate with SAHPRA to oversee vaccine safety. For passive surveillance, we actually use the Med Safety app. And this is where the public and health professionals are encouraged to report any adverse events. And there's also a hotline for COVID-19. The National Immunization Safety Expert Committee, this is a committee that conducts the causality assessment. So here's a diagram pretty much showing how it happens in South Africa, from adverse events detection to notifying and reporting, management, investigation, analysis, and causality assessment, and all the way back to feedback. And this is just a diagram that's showing about how the Med Safety app works to report an adverse event following immunization. And if you go on the NICD, for example, website, you can even look at COVID-19 surveillance reports. And thank you for listening. And of course, protect yourself from coronavirus. Thank you.
Thank you very much. That was Dr. Kulonji on the COVID-19 vaccine safety. You're encouraged to type your questions on the content of the presentations in the question and answer box. And I'm going to ask Dr. Kulonji to go and have a look at the Q&A box and answer some of the questions that are there. Colleagues have already answered some of those questions from the panelist side, but if you look at the answered tab, you may want to add additional comments to questions that are there in the answered tab, the second tab in the Q&A box. Thank you very much again. That was Dr. Kulonji on COVID-19 vaccine safety. So without further delay, I want to now immediately go over to our second speaker. If you could just unshare your slides, Dr. Kulonji, thanks. And then I will, by way of introduction, share with you this particular slide. I'm going to introduce to you our second speaker, Dr. Yamanya Tembo. She's a public health medicine registrar at the University of Cape Town. And prior to her time as a registrar, she was a district health manager in a rural district in Zambia. And a time there was had ignited her interest in public health. During her time as a registrar, she's been involved in COVID-19 outbreak response management and COVID-19 vaccination rollout. She's passionate about health systems strengthening and is excited about the paradigm shift in healthcare that was forced on us by the pandemic. With that very short introduction, I'm going to stop sharing that slide and ask Dr. Tembo to just open a microphone and share her presentation slides with us and proceed on the topic of COVID-19 workplace vaccination program. And while that is happening, a reminder, please just type your questions for the presenters and the content of the presentations in the question and answer box. The chat box is just for general aspects. For those who've asked if this has been recorded, yes, it is, and the links will be made available after the event, probably Monday, given that it's a weekend after our session has ended, as well as for those who want the CPD link and password to register to do the CPD test. And upon the successful conclusion of that test, you'll be issued with a CPD attendance certificate. That will also potentially be only circulated on Monday as well, because we'll be including all the multiple choice questions on the content of our presenters' slides in their test for you. Without further delay, I hand over to Dr. Tembo. And I hand over to you. If you could open your microphone and proceed.
Thank you so much, Ashraf. And good afternoon, everyone. As Ashraf had mentioned, I'll be just presenting some of the experiences about setting up and running a workplace vaccination program, specifically speaking of experiences from the Sisonke 1 vaccination study that happened at Groote Hospital here in the Western Cape. Oh dear. This is going to be the brief outline of what I will discuss. It looks like a lot, but I'm very confident that we will be able to get through it in the 25 minutes allocated. So the COVID vaccination program was rolled out as a major component of the National Department of Health overall COVID-19 response. And this started on the 17th of February when the first COVID vaccines were administered in South Africa. Now, due to the initial limited supply of vaccines, the vaccines had to be rationed using a risk-based approach. And based on that approach, the rollout plan, as shown on the right, had 3 phases that overlapped. The first phase had healthcare workers. The second phase had essential workers and other people who are deemed to be at greater risk. And then the third phase had people who had relatively less risk. And it was decided that the first access be given to healthcare workers with these 2 things that they considered. One was who is a healthcare worker? So it was very important for us to define clearly who we're considering a health worker. And then secondly, we also had to think about who was at greatest risk. So not only within the greater population of South Africa, but also within, the group of health workers, who's at greatest risk because of the way the staggered delivery of the vaccines would come through to each province and each site. And so the rationale was that we had to protect the health system and ensure that it remains responsive and capable of meeting demand for care. And as a result of us choosing to have health workers as our first target population, most of the vaccine centers were initially located in health facilities, but as you know now have now become decentralized. So now, prior to this setup of the vaccination sites, the provinces had to decide who, had to enumerate the number of health workers within its borders, and this was really just to ensure that there was adequate distribution across the provinces. And then also they had to ensure that there was an equitable distribution between public and private facilities alike. And province then had to identify suitable sites for vaccination to occur in. And each vaccination site had to be registered for accreditation and inclusion on the master facility list that was run by the NDOH. Each site also had to appoint a facility representative. And so during this time, there was quite a number of uncertainties. And because of that, we had to rely very much on our planning, on evidence, and on our agility to be able to cope with these changes. One of the changes that was gives you a stark, example of this was just the change from AstraZeneca to the J&J vaccine. If you remember, AstraZeneca, just before the rollout started, was shown to be less effective against the strain that was circulating at the time. And at the same time, luckily, there was this J&J vaccine that was showing very good efficacy against the circulating COVID strain at the time. And so there was this switch to J&J, and this resulted in a few changes. One was obviously the delay in dates for rollout. Then there was also the change to vaccinator training manuals, changes that had to be made to the electronic vaccine data system, which is a system on which we capture all the activities of the vaccination happening. And then it also resulted in changes in processes at the sites, as we now had to work and accommodate researchers. If you remember, this J&J was given to us as part of a implementation research study. And so we had to then shift strategies to accommodate these changes. So, part of, as all of this is going on, we also had to, go about the actual work of categorizing our healthcare workers within our facility to know who among our staff would receive the vaccines first, and who would, who can stand to wait a little bit later. And so based on the advice from the expert advisory committee, both at the province and at National Department of Health, they made this risk scoring that was based on individual vulnerability by age, by which age, greater age is more at risk, individual vulnerability by comorbidities, and there were specific comorbidities that we're looking at that, increased your risk, your adverse outcomes for COVID, such as hypertension and diabetes. Then, risk of exposure of the staff. So just categorizing each staff, what is your risk of exposure for COVID, and then the criticality of the area in which this particular healthcare worker was working it. And specifically, we're looking at places like your emergency centers, your ICUs, your theaters as critical services that, needed to continue running in the healthcare platform. And so this risk categorization was implemented by our line managers within their teams to categorize each of their staff. And all of this was collated into one master file. So once we had categorized our staff, we went about the actual practical work of setting up our vaccination site. And in doing this, we had a lot of assistance and documents that we received from the provincial health office to aid us in this task. And some of the things that we thought about were, is our venue suitable, taking into account COVID restrictions for social distancing, capacity, and adequate airflow? Do we have the equipment that we need and the information technology? So things like your computers, do we have adequate network lines? How much furniture do we need? Do we need, tables, chairs? Where do we place our bins? Resuscitation equipment, and critical as well was cold storage, because we had to ensure that we maintained the cold chain for our vaccines. Then we also thought about, the consumables that we're actually using during the vaccination process. So such as your alcohol hand rub, your cotton buds, your strapping, and then we also put some thought about towards the information material that we would actually need. So this is for staff, all the training materials that they would need, the job aids that would help them to refer to if they come across any challenges, and also the information that would give the people receiving the vaccines. So this included, the leaflets that they would get as they leave the vaccination center. It's the posters that would show them the flow of things at the vaccination site and where to go. We also had to think about handling of waste. And I mean, lucky for us, we are a health facility. And so because of that, we didn't have to put in too many resources into the way we handle our waste, because we already had these systems in place, being a hospital. And so we only had to think about how we are going to ensure that we maintain these processes from the vaccine center to the places where we dispose of our waste, and also critically ensuring that our vials do not end up in the community, because at this time there was quite a lot of risk with perhaps ending up with counterfeit goods being refilled into these empty containers and then possibly sold on the black market. Lastly, we also thought about, the way the flow through the center would occur. And this we did just to ensure that we minimize the cross exposure between, staff as they walk through the facility. And so just ensuring that we had one entrance, one exit, and one direction of flow through the facility. And so this is how we had a mock of how we set up our facility with one entrance and one exit. And the flow through this facility was counterclockwise. And we were lucky in that our site had actually been built especially for the vaccine, the COVID testing. And so it had adequate ventilation, it had extraction. The only thing that we had to add in were a few network points and all the computers, but it was a very adequate space for us to use. As you can see on the top right, that's just some of the cubicles, with staff getting vaccinated. And on the lower left, that's the entrance to the registration area and a pre-vaccination waiting area where the chairs can be seen. So now, before the health workers presented themselves to the vaccine center, there were some processes that they needed to do. Healthcare workers had to register on the electronic data system. And once they registered, they would receive this invitation to participate on the trial based on their eligibility and prioritization. And then they would also get an acceptance, or they would have to accept this invitation and complete a consent form online. And once this was completed, they would then receive a voucher and an invitation to present to a named facility. And in this case, all our staff were coming to British Care. And the health worker was then rostered on an appointment schedule, given a date and a time for when they could come and receive their vaccine. One of the things that we noted during this process was that not everybody in our facility had access to a computer with internet or a smart device that had internet connection. And so we had to think about where we would place these, how we would help these staff to get registered. And so initially we had a computer lab set up where staff could go and get help to register. And later on, we decentralized this computer lab to the front entrance of the vaccine center, so that as they are waiting to enter the vaccine center, they could get help to register on the EVDS system. So the process flow through the vaccine center, once they arrived and they had gotten their voucher, started first at the screening. So when they would come, they would be checked off the appointment list, and they would be screened for their COVID symptoms. And remember, at this point, I also mentioned that at this point is also where they could also get assistance to register on EVDS. Then they would go onto the registration part of the process, where their documentation would be verified by clerks, and the details would be checked on the EVDS records to ensure that these things tallied and everything that was captured correctly. And additionally, and this was an additional point as well, to help staff get registered if we had a long queue building up, say, for instance. Then they would move on to the actual vaccination stations, where they would get screening for medical questions would be asked to them, just to determine whether they have any contraindications to receiving this vaccine. As Dr. Kalonji mentioned, there are some ingredients that some people might be allergic to within the vaccine. So we will try and screen for these people and deal with those people who are presenting with risk accordingly. The vaccinator would then administer the vaccine, and these details would be captured on the EVDS. The health worker would then move on to the observation area where they would wait for 15 minutes. If any adverse events happened, they would be dealt with in that area as it had an adjoining resuscitation area. And if it was felt that there was any more serious intervention needed, they would quickly be rushed off to the emergency center, which luckily for us was right outside the exit doors of the vaccination center. So this is just showing the entrance to our vaccination center and some of the posters and the information that was given to health workers prior to them coming to the vaccine center. This also just showing some of the queues that would build up going all the way up the hill. The top right picture there depicting the research staff receiving the vaccines into the vaccine center and ensuring that cold chain had been maintained, and then also the vaccinator at work. So when we looked at the staff and the training that was needed, we relied heavily on volunteers for our staffing, clinical volunteers and clerical staff. And we also relied heavily on our managers, medical managers, to help us ensure that each of the cubicles had a vaccinator on schedule for the day. And they would manage those schedules. And if there were any gaps, the overall site coordinator, who happened to be myself at the time, was in charge of making sure that those vaccinator slots or administrator slots were filled in. And this was done with the help of other partners, such as the university, UCT. We had volunteers from UCT and from other health facilities within the area. Then we also ensured that all our staff had training, and this was done by training modules that was provided by the NGOH and adapted to be more comprehensive and summarized, because we were cognizant that most of the vaccinators on our staff were already health staff who knew these things, but just needed a bit more focus to thinking about the COVID vaccine. Then we also had visual guides to help our staff, which were pasted at each of the vaccination stations and at the admin stations. And then we also had training for our staff on how to use EVDS and get them, their credentials registered on EVDS so that they were ready to go once the program started. So as part of our preparation, we also had to think of vaccine acceptance and hesitancy. This is because this was a new product on the market, and understandably, there was a lot of anxiety around this product. And so the Provincial Health Office carried out a survey prior to the commencement just to find out what the acceptance levels were. And there's a picture of some of the results that came out of one of the surveys that was done. And it just showed that there was a good level of vaccine acceptance, about 59%, but also quite a high level of vaccine hesitancy. But this isn't to be confused with vaccine anti-vax sentiments. This really just showed us that there were key issues that our staff were not very sure about or had concerns about the vaccine. And because of that, the province then decided to hold vaccine information sessions. But these were mostly online, on online platforms. And remember, not everybody had access to these online platforms. So a multidisciplinary team was formed at Critics Care, and they had local in-person information sessions that ran out throughout the day, every day, from the week prior to the commencement of the vaccines into, I think, 2 to 3 weeks down the line. And the sessions comprised of staff who are working at the hospital, occupational health and safety, IPC, public relations officers, doctors, nurses. And this was phenomenal because it was fellow staff giving advice to their fellow staff. And because of this, these were people who they hold in high esteem, who they could trust, who they felt that they could go to if they had any other further questions. And these people were able to break down the information into simplified language to help them to understand. And as more and more people got vaccinated, with no adverse effects, more and more people were encouraged to get vaccinated. Then during the process, we also had to manage the adverse events. And so for immediate reactions, we had set up a resuscitation area right next to our post-vaccination waiting area, which was fully equipped and manned by 2 nursing staff at every moment. And there were doctors also on duty. We ensured that there was a doctor on duty all the time in the vaccination center if we needed any extra assistance. And then, as I mentioned as well, we were also conveniently located right next to the emergency center where we could quickly rush off a health worker if need be. And then based on what was most likely symptoms for staff, they were advised on what they should do in the event that symptoms happened when they go home. For later onset symptoms, staff also presented themselves to the occupational health staff clinic, and they were usually given about 2 days off. They were referred to the Sisonke help desk because remember, we still had to report these findings to the study arm. And then we also referred them to GPs if we had any concerns about the seriousness of the symptoms. We then also had to report these adverse events, all of them, through the adverse events for following immunizations through to the province. And for the majority of the staff, there were very few side effects. Most of the side effects that were noted, about 81%, were mild to moderate. And only 50 health workers had adverse events that could be categorized as serious or of special interest. Only 12 of them had allergic reactions that could be classified as anaphylaxis reactions. So this is just some of the leaflets that we're giving to the staff. So some of the challenges that we faced were, to do with governance, because it was a very strict governance being held strictly by NGOH. It was sometimes difficult to effect quick changes on the ground because we needed that permission from province through to the National Department of Health. And then also as a direct consequence of that, when you're looking at data management, we couldn't easily access the reports from EBDS. So even though we were giving all this information into the data system, we couldn't pull out reports to help us to change our strategies or ensure that we are reaching all the health workers who are registered on our system. When we looked at infrastructure, some of the challenges were just because of the long queues and the social distancing requirements and the weather at a certain point. It was difficult to have all these health workers in one place. So it was quite a challenge in trying to ensure that we are adequately social distanced within the facility, especially when there was bad weather. Then, we spoke about risk communication in vaccine hesitancy and just the difficulties in trying to address each and every one of the healthcare workers' concerns, particularly that as we were, we were trying to counteract some of the misconceptions. There were new misconceptions coming out almost hourly, it felt like. So it was quite a challenge in that. Then I think specifically when we're looking at risk categorization, when we looked at who is the healthcare worker, it was sometimes difficult to completely define who is a healthcare worker, because at some time the criteria was widened to include any staff that directly or indirectly assisted in improving the health of the health outcomes. And so, do you include people who supply prosthetics to a hospital? So there was a few gray areas that we had to handle and kind of delineate for ourselves within the facility. Then also challenges in just, categorizing the kind of exposure that someone has was quite difficult. Then when you look at the logistics and planning, there were several delays that really just were part of the research component because of the stringencies that were, had to be maintained because it was a research component. And then the fact that we relied heavily on staffing also really, caused a challenge because then we constantly had to ensure that we had adequate staff every morning and fill in those gaps whenever we didn't. Then when looking at the successes, I think I cannot underscore the immense success that having this multidisciplinary team managing the site played into the overall success of the program. We also were able to, we had daily meetings with the provincial health office, and I think our feedback really laid the groundwork for some of the governance arrangements to change and allow flexibility such that when the program rolled out to the greater community, it was much more flexible and easy to implement. Then when we looked at the data management, we had, if you recall, we had issues with getting data back from EVDS. So we had to keep parallel records of the activities on the ground to help us kind of understand what was going on. And then also us enabling the access for those without smartphones or internet connectivity, I think was a great success story. Then I think with the infrastructure, we really lucked out by having this newly built facility that was able to accommodate the needs of a COVID vaccine center, given the restrictions. Then when we look at the risk communication, again, we're looking at those information sessions that were really well received and well run every day, 3 times a day. And again, just the wealth of the multidisciplinary team that came together to actually give these sessions and run these sessions. And then lastly, I think one of the biggest successes was also just this keeping our communication lines open, both internally and externally, which facilitated swift changes and contributed to our low vaccine wastage of less than 1%. And I cannot underscore the impact of this program. I'm sure a lot of you had seen this graphic that was circulating, just showing what we were initially seeing in early September following the vaccine program, of how many people were hospitalized and in high care and on ventilators at Critical Care Hospital. The study that followed, the Sisonke implementation study, also showed that of the individuals who were vaccinated, we were able to prevent 83% of COVID-related deaths and prevent 75% of COVID-related hospital admissions requiring critical or intensive care, and 67% of COVID-related hospitalizations. Thank you so very much for your attention. This is just some of the references that I use that you can look at in your own time to get a bit more understanding of everything that went on. And many thanks to the team that assisted me. Thank you.
Thank you very much. That was Dr. Yamanya Tembo on the topic COVID-19 workplace vaccination program, and giving you significant insight into what has happened there in her experience. So without further delay, I'm going to immediately introduce our next speaker. And oops, I think that's the wrong screen. There we go. And I'm going to introduce Professor Shabir Mahdi, who will be introducing COVID-19 vaccine update for us. Prof is the Dean of the Faculty of Health Sciences and Professor of Vaccinology at the University of Witwatersrand in Johannesburg. He also holds the position of Director of the South African Medical Research Council Vaccines and Infectious Diseases Analytics Research Unit, the VIDA, and is co-director of the African Leadership Initiative for Vaccinology Expertise. That's alive. He is internationally recognized for his research on vaccines against life-threatening diseases in childhood, in pregnant women, and against respiratory diseases, which informed the World Health Organization recommendations on the use of these vaccines in low and middle income settings. Most recently, he led the first 2 COVID-19 vaccine studies being undertaken in Africa, and has been involved in multiple epidemiological studies in COVID-19 in South Africa. He's been widely complimented for his stance on COVID-19 in South Africa, where he's considered a trusted and unafraid voice, a trusted voice, unafraid to speak truth to power. So with those words, I welcome Professor Shabir Mahdi, and I see that he's already sharing his slides. I'm going to ask him to open his microphone and then to proceed with his presentation. Thank you very much, Professor. And there you go. Please go ahead. Thanks.
Thanks, and thanks for this invite. So what I've been requested to do was to give an update on COVID-19 vaccines. And I guess what's of particular importance in the current context in South Africa is the effectiveness of vaccines against Omicron, but also how to move forward in the midst of what has really been a vaccine rollout program that has struggled and certainly has missed almost all of the targets which it initially set out to achieve. But before doing that, I think it's important for us just to reflect as to what the COVID trajectory has been in South Africa, because that will to some extent determine what we can expect moving forward, and should also influence the manner in which we think about continuing rolling out vaccines, and particularly where to actually focus rather than being extremely diffuse and probably achieving very little consequently. Now, one of the major challenges that we have faced on the African continent, and South Africa is no exception, despite it being a country with amongst the highest testing rate across the entire continent, is that compared to many other countries, we have pretty much lagged behind when it comes to the number of tests that have been done per capita. So I'm just using two examples here. That's the United States and United Kingdom. And what we find is that during the course when Omicron wave was circulating in South Africa, when we were reporting 29,000 cases and the United Kingdom were perhaps reporting 50,000 cases, the reality is that it's impossible to make any head-to-head comparisons between those numbers, other than the fact that the United Kingdom has got a larger population by about 20 million. What's even more impressive is that they were doing 20 times more testing than was being done in South Africa. And similarly in the United States, that has got a population size which is 6 times greater than South Africa, when they were documenting 1 million cases of COVID at a peak of the Omicron wave, and South Africa was reporting a maximum of 30,000 cases at a peak of the Omicron wave. Those reports were coming from a country, as I mentioned, that was 6 times larger than South Africa, and that was doing 6 times more testing than South Africa. So given the limited amount of testing that's been done, and like I said, South Africa has performed much better than most other African countries, being one of the reasons why more than 40% of all of the COVID-19 cases documented on the African continent has taken place in South Africa. What we can't do is make any sort of meaningful head-to-head comparisons with other settings where testing rates are very different. Now, what is this actually materialized in? So currently, I think the estimate, and I've stopped looking at the numbers because the numbers are pretty meaningless. I think there's about 3.5 million people that have been documented with COVID-19 in South Africa. But the reality is that after the Omicron wave, we're probably now sitting with 80 to 85% of the population having been infected with the virus at least once, which obviously makes a mockery of continuing reporting on 3.5 million recorded cases, because what we've now come to understand through a series of sero surveys that have been conducted is that the force of infection that has transpired in South Africa has been huge. Despite all of the restrictions that government tried to impose, all of the regulations, some of which are completely obsolete but are ongoing even in this day and age, despite all of those regulations, before the onset of the Omicron wave, and this was a population-based survey in Gauteng, what we identified was that 73%, 7 out of every 10 people in Gauteng, had been infected with the SARS-CoV-2 virus at least once since the start of the COVID pandemic. After the Omicron wave, which was due to a variant that is much more infectious than anything that we had seen earlier, and was causing a large number of breakthrough infections as well as reinfections in people that were vaccinated or people with past infection respectively. After the onset of the Omicron wave, we can now expect 80% to 85% of people to have been infected with the virus at least once in South Africa. And these are data which are not thumb-sucking. This is a repeat of a sero survey that is currently underway in Gauteng, population-based sero survey, where the current average is 85% after the Omicron wave. Now, even before the Omicron wave, in some places in Gauteng, as an example, in the city of Johannesburg, the percentage of people that were testing positive for antibody as a metric for past infection was 85%. Now, Gauteng makes up 25% of the South African population, so roughly about 15 million people. And in Gauteng alone right now, the number of people that have been infected with SARS-CoV-2, 80% of 15 million, you can do the mathematics, and clearly nowhere close to 3.5 million. So 3.5 million at the national level is a massive underestimate. And the number of people that have been infected with the virus up until now in South Africa is in a region of 45 to 50 million. Rather than 3.5 million. So this is important because what we've now come to understand about natural infection, and it is not unique for SARS-CoV-2. This is the way most respiratory viruses evolve over time and why they become more of a nuisance factor eventually, might still cause death, but become more of a nuisance factor is because we have evolution of natural infection-induced immunity. Now, that is not to say that we're encouraging people to go out to be infected, and I'll show you why. Now, in addition to that population-based sero survey, to corroborate those findings was another study that was done by Cheryl Cohen from the NICD, where they did a household transmission study in a rural setting and in an urban setting. And what they documented, again, before the onset of the Omicron wave, was something which was very similar to what we had shown in a cross-sectional population-based sero survey. And that is in urban settings, 75% of people have been infected at least once in these households since the start of the pandemic. And in a rural setting, it was sitting at about 60%. But what you also see quite strikingly here is that the positivity rate from serology is almost identical to the positivity rate for serology and PCR, or PCR, which means that very little additional cases will be documented by PCR compared to what were documented by serology. But more important than that is 85%. Now, this was where serial samples were being collected in the household. 85% of infections that transpired were completely asymptomatic. Now, why is this important? This is important because if you are wanting to, for things such as contact tracing, isolation, and quarantine to work, you need to be able to identify close on to 90% of the infections that are taking place to start off with. When 85% of those infections are asymptomatic and your testing rate is nominal for all intents and purposes, you're effectively not identifying more than 10% of all of the infections that have transpired. And that makes it completely meaningless to continue with contact tracing because you're not identifying the majority of contacts with people that are infected. It makes it meaningless to ask people to go into quarantine because the majority of people that are exposed to someone that are infected don't know they've been infected. And in fact, when it comes to people going into isolation, you can also debate the merits of doing that. Because as I mentioned, only 1 out of every 10 people that are infected are actually going into isolation. So you don't expect the trajectory of the pandemic to be very different than if those 10% of people continued mingling in society. And that is the reality. Now, that such a large percentage of people who are asymptomatic shouldn't come as a surprise because 6 months into the pandemic, there were already reports that 50 to 60% of infections in countries where a lot of testing was taking place, a lot of contact tracing was taking place, 50 to 60% of infections were asymptomatic. In children, that was as high as 80%. Now, what is all these large numbers of infections materializing? It has materialized in evolution of natural infection-induced immunity. And natural infection-induced immunity works pretty well, not only against protecting against severe disease, but as I'll show you, probably works slightly better than even messenger RNA vaccines in protecting against infection due to a new variant. But this massive infection that has taken place came at a huge cost of life. So the number of people that have died of COVID-19 in South Africa is not 99,800 and whatever other unit you want to put to it. The number of people that have died in South Africa to date is closer to 300,000. And these are based on the South African Medical Research Council excess mortality estimates. Now, why do we believe that the majority of this excess mortality are COVID deaths and not something else, some other disease that has been neglected during the course of COVID? And the reason for that is that if you look at the trajectory of the COVID reported death and the trajectory of this excess mortality, they're almost completely in sync, strongly indicating that the main way to attribute, the only disease to really attribute this excess mortality 2 is COVID-19. And this is exactly the same sort of modeling that is usually traditionally used in other settings, including in South Africa, to sort of try to understand, as an example, the number of people that die of influenza illness. So the number of people that have died of COVID-19 in South Africa is closer to 300,000. Now, 300,000 itself is a meaningless number because 300,000 in South Africa might mean something very different compared to 1 million people having died in the United States, which is what they're reaching. Does that mean the United States has done worse in South Africa? And the answer is no. And the reason for that, like I mentioned, the United States population is 6 times greater. They've done more testing. And in fact, the majority of people that have died is actually accounted for or recorded. And the recorded deaths and excess mortality are almost identical in the United States, or maybe a 15-20% difference. Now, the better way to interpret the data is to look at the number of deaths per 100,000. And in South Africa, we're sitting at 500 per 100,000. That places South Africa firmly amongst the top 10 countries with the highest COVID mortality rate globally. If the Northern Cape was a country of its own, sitting at close to 700 per 100,000 deaths due to COVID-19, the Northern Cape would be amongst the top 3 countries with the highest COVID-19 mortality rate globally. So for all purposes, South Africa has performed dismally in protecting against infections and hasn't done well in protecting lives being lost due to COVID-19. Now, part of the reason for that, unfortunately, is a consequence of what has been a disastrous rollout of COVID-19 vaccines. And we should stop pretending that we've made significant progress because, like I said, we haven't. And the reason for this, unfortunately, goes back to January of 2021. On the 2nd of January, a group of scientists wrote this article in a Daily Maverick, which clearly pointed out that whilst many other high-income countries, as well as many African countries, were beginning to deploy COVID-19 vaccines into the immunization program, and these were vaccines that started to be deployed after those really fascinating data came through in November of 2020, South Africa yet didn't seem to have a plan. And in fact, if anything, there were people, senior officials, that were downplaying the prospects for the availability and usefulness of COVID-19 vaccines in South Africa. The government was called upon to basically indicate what exactly its plan was. And Zapiro pretty much summed up what the government's rollout plan was in the beginning of January 2021. And that it was completely absent. Overnight, a plan was sort of cooked up. Government indicated a few days later that they were going to vaccinate 40 million people by the end of 2021, 40 million people by the end of 2021. And then they came up with this roadmap, which I think the previous speaker showed, about which groups were going to be prioritized and which groups were going to be completed by when. I think people above the age of 60 were meant to have been completed by about May of 2021 already. And there were all of these ambitions, aspirational goals, which clearly were going to fail for a number of reasons, including the reality that there was very little preparation. In addition to no vaccine having been procured at that point in time, there was simply very little preparation for the deployment of these vaccines. And then there was this notion that South Africa is somehow unique and was going to implement some electronic vaccine data system, which was going to shepherd people to go to a specific place to be vaccinated at a specific time on a specific date. Now, that is somewhat oblivious to the realities that most South Africans live under. And that is the vast majority of them don't have access to be able to actually register on an EVDS site, let alone plan to be at a place where they're expected to be on a given time and a given date without being provided resources to do so. So what materialized? What materialized is that eventually government was able to get 1.5 million doses of vaccine from Serum Institute of India. It arrived on the 1st of February 2021 amongst a lot of fanfare. Unfortunately, we were at the same time doing a study of the AstraZeneca vaccine, and we actually ended up releasing our results the very same night that these vaccines arrived in South Africa. And disappointingly, what we showed is that AstraZeneca vaccine didn't protect against the beta variant, which was then dominating the COVID wave in South Africa. What we showed effectively is that there was about a 10%, so basically no efficacy against the beta variant. but this was against mild to moderate COVID-19. The study wasn't designed to look at severe disease. We didn't have the right population group to look at severe disease, but we showed that the vaccine doesn't protect against mild to moderate COVID-19. Now, that didn't come as a surprise, because before we did an unblinding of the study, we also had data from the NICD, from Penny Moore's lab, from Alex Siegel's lab, which convincingly showed that a beta variant was relatively antibody evasive to the neutralizing activity that was induced by the vaccines. So it didn't come as too much of a surprise that in the absence of neutralizing activity that is induced by the AstraZeneca vaccine against the beta variant, the vaccine is not going to protect against mild to moderate COVID-19. But at the same time, what we showed in the same study was that although the antibody didn't show neutralizing activity against the beta variant, and that is antibody induced by the vaccine induced T-cell immunity, the CD4 and the CD8 responses were largely preserved. 76 of the 87 T-cell responses that's induced by the AstraZeneca vaccine were largely preserved, including most of the natural killer cell responses. So at that stage, we were already seeing a disconnect of how vaccines protect against infection as opposed to severe disease. And consistently, all of the studies were showing greater protection against severe disease compared to how well vaccines were protecting against infection and mild disease. Now, based on these data, coupled with animal experiments that were being done at the same time, where mice were basically given the AstraZeneca vaccine and then challenged either with the alpha variant or the beta variant. And what they showed in these animal model studies, or they were challenged with a control, which basically means they were not vaccinated, but challenged with they were controls not vaccinated, but challenged with the same variants. And what they showed is that the mice that were vaccinated, when they were challenged with the alpha and the beta variant, their lungs remained completely healthy, macroscopically and microscopically. Mice that didn't receive AstraZeneca vaccine, their lungs were pretty much destroyed after being challenged for the alpha as well as for the beta variant. So these were data that were now accumulating, indicating that although the AstraZeneca vaccine wouldn't protect against infection and mild COVID, it could still protect against severe disease. Now, based on this, WHO made a recommendation that there was indirect evidence which is compatible with the protection against severe COVID-19 for the AstraZeneca vaccine. And even those countries where the beta variant was circulating, they should still continue using the AstraZeneca vaccine in those countries. Why? Because not we were trying to prevent infections, but because vaccines are really designed and have always been designed mainly to prevent severe disease and protect people from dying. When vaccines protect against infection, that's a welcome bonus. And that allows us to bring about a reduction in transmission of the virus much more effectively. But that's not what vaccines are developed for. So WHO made this recommendation, and this recommendation was subsequently supported by a study from Canada, which basically showed that even a single dose of the AstraZeneca vaccine, or 2 doses, provided 80 to 90% protection against hospitalization and death due to the beta or the gamma variant. Against the Delta variant, the AstraZeneca vaccines, that's the AstraZeneca vaccine, a single dose here. Against the Delta variant, basically AstraZeneca as well as the messenger RNA vaccines, you've got close on to 90% protection against hospitalization and death. So South Africa used the AstraZeneca vaccine rather than selling it off to other African countries where the beta variant was also circulating, with that 1.5 million doses of vaccine. Yes, I'll show you. We probably would have saved 20,000 lives, 20,000 people from dying above the age of 50, had they received only a single dose of the AstraZeneca vaccine, which conferred 80% protection against hospitalization or death due to the beta variant. But as it happened, the beta variant wasn't a problem by the time we started to roll out our vaccines, not in February. February was really for healthcare workers, and that wasn't a rollout of vaccines. The vaccine rollout really only started around about June, very much at a time when the Delta variant had arisen and pretty much had replaced the beta variant. And as I showed you, as it happened, the AstraZeneca vaccine and every other vaccine works extremely well in protecting against the Delta variant. So not only did we go against WHO recommendation for a beta variant, but had we not been stubborn and listened to WHO, we actually would have introduced a vaccine and we would actually have had protection against the Delta variant. And we might just well have had more vaccines coming in from India rather than only the 1.5 million, which would have accelerated the start of the vaccine program. Where did we sit when it came to the Omicron variants? As you've seen here, and as all of us know, on the 25th of November, there was an announcement that Omicron wave is a new variant that evolved. And very soon we saw the Omicron variant very much displace the Delta variant in South Africa. Now, in Gauteng, at the start of the Omicron wave, on the 25th of November, only 1/3 of the population had received at least a single dose of a COVID-19 vaccine. In people above the age of 50, rather than 90% of them having been vaccinated, it's set to 60%. In the 12 to 18 year age group, 11%, 18 to 50 year age group, about 1/3 of those. So clearly we were nowhere on track, almost by the end of 2021, to get anywhere close to 70% of the population being vaccinated, which was what the government had indicated its target was in January of 2021. Now, despite that, as I mentioned, we had already shown now, before the onset of the Omicron wave in Gauteng, that even unvaccinated individuals, including children as young as 12 to 18, 73% of them were seropositive, meaning that they were previously infected with the virus. Adults above the age of 18, 70% of those that were unvaccinated were seropositive. The vaccinated individuals, 93% of them were seropositive, which is not a surprise because vaccines are going to obviously result in an antibody response. And this antibody response, although the antibody might not be a metric as to whether people are protected against infection, is a reasonable proxy as to the extent of underlying T-cell immunity that exists, which will protect against severe disease and death. What was the consequence of all of this? And I'm just going to take you briefly through this here. So this basically is again for Gauteng, and for most of the other provinces, it would plot almost in a very similar manner. And what it shows you in the dotted orange is the number of infections that are reported, in blue the number of hospitalizations that are documented with the virus, including incidental infections. In black is the number of people that have died of COVID-19. And particularly here, I'm showing the excess mortality estimates. In solid orange is the number of people that have been recorded to have died of COVID-19. The percentage indicates what percentage of those events occurred in a particular wave since the start of the pandemic, and the number in parentheses indicates the incidence per 100,000. And what we see happening, despite a modest uptake of vaccine in Gauteng, is a massive decoupling of infections and severe disease and death. So the Omicron wave contributed to only 10% of all of the infections that occurred in Gauteng since the start of the pandemic, or 10% of all hospitalizations, 20% of all infections since the start of the pandemic. And only 3% of all of the people that died of COVID-19 in Gauteng since the start of the pandemic occurred during the course of the Omicron wave. In contrast, before we started to roll out COVID-19 vaccines and the missed opportunity of introducing vaccine at the end of the Beta variant wave, the Delta variant wave was responsible for 44% of all of the hospitalizations that had transpired since the start of the pandemic, and half of the people that died in Gauteng were deaths that occurred during the course of the Delta variant wave. 50% of all of the deaths that occurred since the start of the pandemic occurred during a single wave when there was almost absolutely no protection except for healthcare workers through vaccination. Because South Africa didn't roll out a vaccine, went against WHO policy, didn't procure vaccine early enough, didn't have a plan in place. And this is the human cost of having really messed up with the vaccine rollout program. So these data are very similar in a 50 to 59 year age group. Only 2% of all of the deaths that has occurred in this age group, including people above the age of 60, occurred during the course of the Omicron wave. And the reason why this age group is especially important is because 80% of all of the COVID-19 deaths before the onset of the Omicron wave had occurred in people above the age of 50 in South Africa. So we've seen a massive decoupling of infections. 53% of all of the deaths in this age group occurred again during the course of the Delta variant wave. Children 5 to 19, where there was a modest uptake of vaccine, again, we see a very similar sort of a picture. Their mortality rate was always very low in the region of 1 per 100,000. 17% of all of the deaths in children 5 to 19 years, where there was very little vaccine uptake, occurred during the course of the Omicron wave, compared with 47% having occurred during the course of the Delta variant wave. But important in children is that this incidence for death, 1 per 100,000, is substantially lower than the incidence that's observed in people above 50 to 59, as an example, where even during the course of the Omicron wave, it's still sitting at 9 per 100,000 rather than 1 per 100,000, which has always been the case in children under the age of 18. Now, what do we know about vaccines and Omicron? So these were data published from South Africa at an early stage after Omicron arose, published from a discovery database, which not unsurprisingly didn't show it showed that the vaccines were less effective against Omicron than the Delta variant wave. The protection against effectiveness of 2 doses of the Pfizer vaccine was sitting at 33% compared with 80% due to the Delta variant wave. And the longer ago that people received their second dose of vaccine, the lower the effectiveness, because in addition to Omicron being antibody evasive, there was also waning of antibodies. So people that had received their second dose 3 to 4 months before the onset of Omicron, vaccine effectiveness there only was around about 25%. Now, that didn't come as a surprise, as I mentioned, because what we had seen from the laboratory, even before the data became available, is that this is looking at neutralizing antibody activity in people that received 2 doses of Pfizer or 2 doses of AstraZeneca, 4 to 6 months later, 1 month later, 2 doses of Moderna, and then 2 doses of Pfizer. But all of the studies, what they show is that after 2 doses of vaccine, when you look at the percentage of the number of people that have antibody activity against Omicron, is almost 0. 1 out of 10, 0 out of 10. And for Pfizer after 2 doses, slightly better, but that was because it's 1 month after the second dose. So this tells us that Omicron is antibody evasive, and we don't expect 1 or 2 doses of vaccine to do too much, and less so if the vaccines were given some time ago. But in addition to that, people with past infection due to the alpha variant, beta variant, delta variant, they too showed very little antibody activity against Omicron. But where it differs is people that might have had a heterologous approach, where they've had a combination of AstraZeneca followed by Pfizer, or this could be Johnson & Johnson followed by Pfizer, where that heterologous approach induces a much more robust antibody response that is actually able to overcome some of the antibody evasiveness of Omicron. 14 out of 20 still had activity if they had this heterologous vaccine approach, which was something that we were advocating for healthcare workers a long, long time ago, more than 6 months ago. Now, although Omicron is antibody evasive, what we also see is that Omicron is still unable to fully evade T-cell immunity. And in fact, the majority of T-cell responses still are relatively preserved, including 75 to 83% of the different CD8 responses. When we talk of T-cell responses, there's multiple different epitopes that are actually targeted to the T-cell responses, much more diverse than epitopes that are induced in neutralizing antibody responses. And the vast majority of the T-cell responses remain relatively intact. And that's the reason why, although the vaccines might not protect against infection in mild COVID, we find that with a discovery database, 2 doses of Pfizer vaccine still at 70% protection against hospitalization due to Omicron, although that was lower compared with effectiveness against the Delta variant. Now, these data from discovery were very much replicated in another study from Qatar that was just published. It's actually online, it's not yet been published. And quickly, what they've shown is that people that received 2 doses of the Pfizer vaccine or 2 doses of the Moderna vaccine, if the second dose was given 1 to 3 months before the onset of Omicron, 45% protection. If the second dose was given more than 7 months ago, no protection against Omicron. The same thing for the Pfizer vaccine, the same thing for the Moderna vaccine. So antibody, in addition to Omicron being antibody evasive, there's also the waning of the antibody, and effectively you don't have much protection 6 months after having been vaccinated against infection. But once you give a booster dose of vaccine, that effectiveness against infection and mild COVID now goes back up to in the region of between 40 and 50%, for both of the messenger RNA vaccines. But what you see for severe disease, a very different picture, where, yes, as was shown in South Africa for the Pfizer vaccine, protection after 2 doses, even 7 months after the second dose, is 78%. By giving a booster dose, you can take that up to 90%. With Moderna, in fact, there isn't too much to be gained. Perhaps there's overlaps of the confidence intervals, but with a third dose of vaccine, you probably are getting additional protection against severe disease. Now, this is not to say that we need to do the same in South Africa. because unlike South Africa, Qatar hasn't had a high force of past infection. And that high force of past infection is important because a hybrid immunity probably is the most potent type of immunity that you can develop, not only against protection for severe disease, but also when it comes to protecting against infection and mild COVID. And the vast majority of people in South Africa that have been vaccinated, in fact, have got hybrid immunity because, like I mentioned, 80% of people have now been infected with the virus at least once. So my last slide is just how good is past infection in protecting against infection, symptomatic illness due to Omicron compared to with messenger RNA vaccines. And this is data again from Qatar, and this is against Omicron. And what they basically show is that, in fact, past infection, this is Omicron as well as other variants. And what they show that past infection, in fact, works better than 2 doses of the messenger RNA vaccines in protecting against symptomatic illness, not against severe disease. Against severe disease, they probably work the same. But past infection works as well, if not better, in protecting against symptomatic illness in people that have received 2 doses of the messenger RNA vaccines. Roughly about 50% more protection from past infection. And this is data that has just come out literally in this week. So it's in preprint, it's not been peer reviewed, but it's got important implications because we cannot pretend that past infection is not important. And these are the data we need to use to inform how we go about continuing with our vaccine rollout, rather than just aspiring to some magic number which is meaningless. Past infection, as shown here, irrespective of whether people were vaccinated first and then developed infection or had infection and then vaccinated, as you can see, when it comes to antibody persistence in terms of neutralizing activity against Omicron, almost all people with hybrid immunity actually also still have protection from symptomatic infection due to Omicron. So hybrid immunity is a major potent driver in protecting people from being infected. So past infection doesn't protect you completely against infection due to Omicron, but certainly people that have hybrid immunity might well be protected against infection and probably be less likely to transmit the virus. And that is where the entire discussion on mandatory vaccination has now fallen into inadvertently in South Africa because we didn't plan for 80% of the population to be infected. It's come at a massive cost of life, but we are where we are. And this hybrid immunity results in us being a very different situation when it comes to what we can expect moving forward. So in conclusion, the COVID-19 regulations in South Africa unfortunately failed in preventing spread of the virus. Had they worked, we wouldn't have had, and this includes everything, including wearing of face masks. 99% of South Africans don't wear the right type of face mask, don't wear it correctly, don't expect it to prevent infections, don't expect it that you're going to prevent transmission into the environment. It's not going to happen. If face masks were working in South Africa, we wouldn't have had 80%, 90% of the population been infected with the virus at least once. And the same thing goes for physical distancing. The one measure that does work is ventilation and ventilation and ventilation. And that's the reason why the risk of being infected with the virus, 80% of infections occur because of indoor gatherings, either directly or indirectly, and especially indoor gatherings in poorly ventilated spaces. So with that, I mean, the fact that government and Department of Health are still wanting to keep regulations in a notifiable medical condition to try to prevent infections in South Africa clearly demonstrates a lack of understanding as to what has transpired and what utility those regulations have served up until now, because it hasn't protected against infections. As I mentioned, we need to get as many people vaccinated as possible. But at the same time, if we're wanting to benefit from vaccines, before the next resurgence is upon us, it's not about vaccinating 10 million or 20 million people. If we get more than 90% of people above the age of 50 vaccinated, that target alone will result in much fewer deaths occurring in South Africa than reaching some arbitrary target of 30 million doses or 40 million people being vaccinated. The focus needs to be right now, and it always should have been, on ensuring that we get more than 90% of people above the age of 50 vaccinated, rather than just changing to younger age groups simply because we weren't getting enough uptake in the greater than 50-year age group. And that speaks to the issue of logistics, planning, deployment, advocacy, communication. Last but not least, yes, we are going to experience another resurgence. There will be more likely other variants, but the reality is that those variants are not going to perform any differently from earlier variants when it comes to how well vaccines protect, as well as how well past infection protects against severe disease. Because if a new variant is able to evade a T-cell immunity, then we're not dealing with COVID-19 any longer. In that case, we'll be dealing with COVID 2022 or COVID 2023, because that will be a very different type of virus and will take us back to March of 2020. So thanks for your attention and opportunity.
Thank you. That was Professor Shabir Mahdi, the Dean of the Faculty of Health Sciences, Professor of Vaccinology at the University of Witwatersrand, on the topic COVID-19 vaccine update. Prof, can you just come back on, because I think there's a few quick questions that I think we must direct to you immediately. It relates to, I think, the fifth wave. It relates to, if I can just quickly, I took a quick screenshot also of some of those questions there. So the one question by Savarani Mudli is, hi, can the NIH, well, can you, Prof, share more information on the predictions of the fifth wave of COVID-19? With winter fast approaching, we want to help our workforce and their families to prepare, as not everyone is in each family is vaccinated, or perhaps might have been positive before. Thus, if a person becomes COVID-19 positive, he or she can spread the virus to other family members. What is your advice on that, Prof?
Yeah, like I said, a resurgence is very likely to occur, probably in about 2 months or so. And at the same time, it might well be another variant, not Omicron. It might be going back to Delta, or it might be something else. But again, it's no longer about preventing infections. And I think it's a mindset shift that we need to undergo. We don't have the tools available to us with vaccines to prevent infections. We don't have the tools available to us to prevent infections, period, in the South African context. Non-pharmacological interventions, like I said, had they worked, wouldn't have resulted in 75–80% of people being infected with the virus at least once, had they worked. And we need to accept that to be able to move on. The focus is no longer about preventing infections. If we continue harping on that, we'll continue doing more harm than any good. Harm to the economy, harm to the livelihoods of people, harm to children that are being deprived of schools. We can't afford that to continue. So it has to be about protection against severe disease. And like I said at the end, it's all about ensuring that we got maximum protection in those individuals that are most likely to end up in hospital or die of COVID-19. In South Africa, that is anyone that has got an underlying medical condition, or especially using it as a proxy, people above the age of 50. But under the age of 50, someone with hypertension, obesity, they're included in that category. And the focus has to be about getting more than 90% of those people vaccinated, rather than pretending that we've got the vaccines at place that is going to be able to assist us in preventing infections, because the next variant might be even more antibody evasive than Omicron, which again means that vaccines are going to play very little role in protecting against infection, but those vaccines still play an important role in protecting against severe disease. Given how we're struggling with our vaccine program, and one of the challenges and reasons we struggle is we're so diffuse in our attention that we can't focus. And we have to focus to get more than 90% above people above the age of 50 vaccinated. So I'm not concerned that children have not been vaccinated. As I showed you, the chances of children dying of COVID-19 is nominal. And the reality is that right now, children have developed immunity against severe disease, unfortunately by virtue of having been infected with the virus in large numbers already.
Thank you, Prof. Can I just follow up and also ask our other two guests to step in after your initial response on this? With regard to workplace specific, I think I just want to get it out there again. There we go. So there was a specific question related to the workplace, and I just slipped it off the screen. There we go. A question for Professor Mahdi. There are workplaces enforcing mandatory vaccination. Given the high immunity in South Africa and the inability of the vaccine to completely stop transmission, what is the rationale to have mandatory vaccination in workplaces? Surely it is more important to vaccinate high-risk groups, which I think you've just indicated again. How does the vaccine actually make the workplace safer? Over to you.
Yeah, I think that's a really important question, and we need to reflect on it, because at a time when those mandatory vaccination policies were developed, it was under the notion that the COVID vaccines that we had reduced transmission as well as protected against infection. And that means that you bring about indirect protection of others because you're unlikely to transmit it onward to others. So that is when this mandated vaccination was conceptualized. Things have moved on since then. We know that vaccines don't work too well against infection. Vaccines, despite not protecting against infection, might still play a role in terms of reducing transmission, because the T-cell immunity is probably bringing about quicker viral control, and consequently, that person that is infected is less likely to transmit than someone that hasn't been previously developed immunity, T-cell immunity. But the point is, with 80% of the population now having been infected and also having developed this T-cell immunity, is an additional reason to continue with mandatory vaccination. Because, like I said, natural infection induces a much more potent T-cell immunity than vaccines do. And the answer to that is, I showed it in the last slide, and I might have been too fast on it, is that this hybrid immunity results in a magnitude of antibody response, which is very, very different, much higher than vaccine-only induced immunity or natural infection-only induced immunity. And consequently, people with hybrid immunity are more protected against being infected with the virus. And if you're more protected against being infected with the virus, you're less likely to transmit the virus. So there's still a case to be made, I believe, especially where people are gathering in large numbers on a frequent basis indoors for mandatory vaccination policy if we are wanting to continue reducing infections and preventing it. I'm not saying that we should allow infections to go rabid. That's not what I'm saying. But we can't focus in preventing infections at the expense of not meeting our targets or protecting against severe disease. That is what I'm saying. But where opportunity exists to prevent infections, we still need to try to make use of whatever tools we have, despite them not being the perfect tools, to try to bring down that infection rate.
Thank you, Prof. So to Dr. Kolonji and Dr. Tembo, there's been questions in this chat as in our Q&A as well as our session yesterday that dealt with ergonomics around the question of workplace screening of workers. Have you got any specific response and advice with regard to how that might be important to consider your own inputs and Prof's inputs, as well as the shifts in terms of the announcements, the official announcements, and the expected changes in regulation and guidelines? Maybe I could, whoever opens the microphone first between Dr. Kalonji and Dr. Tembo, if you could just give some comment on the issue of workplace screening and your experience. Maybe I should start with yeah, go ahead, Dr. Tembo.
Yeah, so I think, you know, given the some of Prof. Shabir Madi's you know, inputs, it's really, I think workplace screening is becoming more and more futile, and we should, again, like, our focus should be in trying to prevent the you know, the severe illnesses and the deaths and the overwhelming of the health system. And, you know, and I think this is kind of where the government is going towards in some of its advice and, you know, and the new regulations that are coming forward following, you know, that we're anticipating following the end of the disaster management regulations. So yeah, I think that's it won't be, it's not useful, and it's just very time consuming. And for the most part, I think you know, the use of those workplace screening or, you know, forms has been variable and not as useful. Usually the contact tracers, whoever it is, would start afresh and, you know, start taking new contact details from you know, the staff who are sick. So yeah, I think just given that we're now kind of, you know, moving our focus towards, you know, trying to prevent severity of illness rather than trying to stop the transmission, which we have failed to do, I think it would be wise to, you know, abandon those workplace you know, logs, so to speak.
Thank you for that. Dr. Kalonji, from your point of view, you've seen the questions in the question and answer box, and you've answered some of them there. What are some of the questions that you've seen, and based on the discussions of your two colleagues as well, are the sort of key take-home messages that you want to share with the audience here in our webinar? Dr. Kalonji, if you could open your microphone. There we go. Please proceed.
Thank you. I think one of the questions that I saw was, what stage is South Africa into regarding the immunization program? And I think after what Prof discussed is the fact that we're probably still in vaccine coverage, because the vaccine coverage is still quite low, which on that graph is around a stage 2. And then there's also mixed feelings about vaccines in terms of the public opinion. So, you know, I would think that we were probably between a stage 2 and 3 in terms of vaccine. There were some questions about innate and adaptive immunity, if there were studies that looked at it. And what I did find is that I did look it up and found that there are studies that were specific to innate and adaptive immunity. And I've just put the links in the chat for anyone who's interested in looking at those questions. But I do agree with both of the panelists that we should actually be focusing on the severity. We've had a few cases at work, and every time we did a bit of an outbreak investigation and we looked at the logs and the temperatures were normal, the logs said no symptoms because it either happens over the weekend or people feel unwell in the evening, and by then they've already been at work through the entire day. So stopping the transmission of infections has actually been quite difficult, but I do agree that maybe the focus should be on the severity of disease rather. Thank you.
Thank you for that. If any of you want to jump in there, please just open your microphone and proceed. If you could repost those links. Go ahead, Prof.
I mean, I'm going to jump in, and unfortunately I need to drop off in 2 minutes. And I think I owe it to Jonathan Erickson, who's posted a whole lot of comments. And I don't want to come across as pretending that we've not seen those comments, and I'm going to respond to it. And it's really unfortunate in this day and age, we've got people still claiming vaccines to be experimental vaccines, which is complete rubbish. So I'm not even going to try to respond to that. The amount of efficacy and safety data that's been generated on vaccines, and the fact that it is licensed in a number of countries, doesn't make it experimental. Vaccinated employees, even in the present context of high force of past infection, are not equally as infectious as unvaccinated colleagues. Unfortunately, you might have missed the last part of my presentation, which clearly indicated that hybrid immunity is much more potent and actually more able to protect people against infection, resulting in them being less infectious. So even in settings where there's been a high force of past infection, vaccinated employees are less likely to transmit the virus than an unvaccinated individual. When it comes to the next question, vaccines have not been proven to be as effective as originally thought. We need to stop manipulating wording. Vaccines were not as effective against Omicron compared against a wild-type virus, because the vaccines were actually developed against a wild-type virus. So we don't expect it to be equally effective against variants, which have become antibody evasive. But as has been shown by the data, these vaccines perform phenomenally well when it comes to protecting against severe disease and death, which is what matters most. In addition to which, the whole issue of side effects of vaccines, which have laid bare recently, clearly demonstrates a complete inability to understand the difference between what are adverse events, which is any medical event that occurs after a person has been vaccinated, irrespective of what the cause of that medical event is, as opposed to a side effect, which is a confirmation that the adverse event was a consequence of vaccination. What was released by the FDA was a whole listing of adverse events and not side effects of vaccines. So again, poor understanding of what was actually released in the public domain. The CDC decided against the use of the Johnson & Johnson vaccine because it's spoiled for choice. The chances of the Johnson & Johnson vaccine causing severe adverse events, serious adverse events, is in a region of 1 out of every 250,000. The chances of dying of a lightning strike in South Africa is higher than the chances of dying of a serious adverse event due to the Johnson & Johnson vaccine. Gates did not say that South Africa and other African countries have reached herd immunity. Herd immunity is a concept that I said 9 months ago we should stop speaking about, because it's never going to materialize in South Africa. What the Gates said recently is that countries in our African continent have had a high force of past infection, and consequently, they've developed high levels of protection against severe disease and death. Reports of side effects met with disdain and ridicule. Yes, we're not talking of side effects, and the reports of the adverse events being attributed to vaccines when they're not should be met with disdain and ridicule. So with all respect to Jonathan Erickson, I think if you're wanting to deal with these issues, then you're needing to deal with these issues in a scientific manner rather than rehashing a whole lot of misinformation that comes from social media.
Thank you very much for that, Prof.
For that, I'm going to need to sign off. I got another call now.
Yes, thank you very much for joining us today. And so as we are in the last minute or two of this webinar, over to Dr. Kalonji and Dr. Tembo, your final comments. We're talking about COVID-19 vaccines in the workplace. We've seen the importance of science. We've seen the importance of speedily sharing knowledge, and expediting the results that may not have yet been fully published in the proper sense, but is made available and can be considered by the experts in the different countries. I mean, I regard our three guests as experts, and so I'll ask to experts online. What are your take-home messages to our audience, particularly those who are responsible for ensuring that our workplaces remain safe places, that prevention within the workplace and workplace preparedness around potentially future issues around biological hazards, viruses, and so on, pandemics of this nature? Over to you. Dr. Lonji, I'll start with you. I see that you are visible, and your microphone, if you could open that, and then I'll end with Dr. Tembo.
Thank you again. Now, obviously, my workplace is a little bit different because we're right in the middle of clinical trials, and we do COVID trials, so we do get information a little bit earlier. But we do focus on clear communication with our staff. That is what's very important. We always talk about risk versus benefits. As soon as there are guidelines that are out there, we communicate. But what we also make sure is that we follow the science, we follow the evidence. I think experts are experts for a reason. And for us, that is actually quite important. I tell people you wouldn't take your car to your neighbor, you would take it to a leadership. I think it's the same thing when it comes to the science around COVID-19 vaccines. We follow the policies as much as we can, and we make the changes when is required. But what's really important is that we do focus on the evidence. If we need to move on looking at severity, then that is what we would do, and we try to be as compliant as possible. But again, we work with COVID every single day, so our situation may be a little bit different to an environment where it's maybe corporate and not part of the research environment. Thank you.
Thank you very much. And then Dr. Kalonji, your side?
Hi. Yeah, so I think for employees, for employers rather, I think one of the things that COVID has taught us is really just the amount of effort and work and thought that we need to put into ensuring that our workplaces are safe. And it's not just like your ergonomics and the usual things. You're also thinking now about communicable diseases, and what is it that you have to ensure within your workspace. What measures do you have to put in within your workspace to ensure that something like this, the fallout of something like this happening again is limited, because this particular scenario, COVID-19 pandemic, was really a ticking time bomb. We knew that at some point something like as big as this is going to happen, and because we haven't put in systems in place, the likelihood that this will happen again is quite huge. So I think, I think even for the NIOH, a lot of the thinking that goes around, the regulations that are in place, the processes in your workplace for occupational health and safety really need to be tightened, and as well as thinking about things like vaccines and how to get your staff to be more amenable to things like that. Yeah, thank you so much.
Well, thank you very much. And before I thank our guest speakers and thank all those participants, we had about, I think, 940 people. At one point, I saw that number in this particular webinar, which is great for us Friday afternoon in particular. So you will see on the screen at the moment, I just want to check with colleagues, can you see the save the date there, Dr. Tembo, Dr. Kalonji, can you see that?
Yes, yes.
Oh, great. So I just want to bring everybody's attention to a very important centenary webinar where we will have reached the 100th COVID-19 webinar since March 2020. It is scheduled for the 21st of April, as you'll see there, starting at 9:25 until approximately 10 to 3 in the afternoon. And we have quite a significant list of guests that we will have on that particular day. And it's going to take me too long to now get that particular program for you to share, but we will be sending out the program once it's completely finalized. It includes the CEO of the NHLS, including the Executive Director of the NIH, Dr. Spo Khalamono, and then it includes other NHLS besides Dr. Kami Chetty, the CEO of the NHLS, Professor Kuleko Mlisana from the NHLS, and then Professor Salim Abdul Karim, Dr. Ivan Ivanov of the World Health Organization, and then Dr. Barry Kisnasamy, who's the chairperson of the COVID-19 Workstream Committee under the National Department of Health. We have a representative from the Department of Employment and Labor, as well as the DME, the mining one, the mining sector. And then we have other very important guests, including academics like Professor Amajibe and Professor Rajan Naidu. I'm going to leave it at that. And on the next occasion, I will share more details of the program, but that's the save the date. You would probably by now have received this email from us if you're on our mailing list on the 21st of March. I'll stop sharing that now and then just say a big thanks to our two speakers that are remaining. That is Dr. Dishiki Kalonji, who is a public health medicine specialist, CRS leader and principal investigator of the South African Medical Research Council's HPRU unit. Thank you very much, Doctor, for making the time, preparing and coming to share your information with us today. And then Dr. Yamanya Tembo, who you also see there on the screen, who dealt with COVID-19 workplace vaccination programs. Thanks for sharing all your practical experience there at Groote Schuur Hospital in Cape Town. And she's a public health medicine registrar at the University of Cape Town, in the also associated and with the Western Cape Department of Health. Thank you very much, Dr. Tembo. Thank you very much, Dr. Kalonji. And in his absence, to Professor Shabir Madi, who has raised very critical questions and yes, challenging us and challenging, obviously, as he says, the authorities to ensure that we sharpen our approach, we sharpen the strategy, and we sharpen the implementation of resources where it is most effective. I say thank you very much to all of you. Have an enjoyable weekend. We will only complete administration for this webinar on Monday, so do not expect any certificates and CPD links and so on soon, as my colleagues who provide support around this matters have left the building. So take care and stay safe and enjoy your weekend, and we'll see you on the next webinar here at the NIH's COVID-19 webinar series. Thank you very much, everybody. Goodbye.
Thanks. Goodbye.